Welcome to ARSI Canada. Let's have a walk around. So here we enter into our lab. This is the nice little reception area. Then we have our admin office on the right. Normally, this is our office, admin, and finance. On our right, we have our well-equipped boardroom. Our kitchen on the left. Small but effective. This is the place where our scientists used to sit, make their reports. Introducing Charlie, he's our lead scientist. Hey
Hello.
Charlie, say hi.
Hi, I'm Charlie Haydikin. I'm the lead scientist here at ARSI Canada.
Charlie, do you mind to show us our lab?
My pleasure.
Have a tour.
Okay, the lab is this way.
I'll be glad to follow you. This looks the changing area, right?
Yeah, this is the changing area where we put on our lab coats and goggles.
Great.
Okay, come on in.
Thank you.
So we'll start on the left side. This is our synthesis area where we do all our synthetic work. We have an extra rotovap here. So we have one rotovap in a fume hood already. And then we have a water purification system, ultrasonic bath and cleaner, and just some PI water where we do all our cleaning.
Great.
Here we have our safety shower. Of course, we have to have a safety shower and an eye wash station. So we are all up for safety protocols. We have an ice maker, and then we have a combi flash, which is another brand of the Biotech. So it's basically we can do high pressure column chromatography here. We can do regular phase or reverse phase depending on the cartridges we use. And we have all of our different solvents and our waste collection and everything we need here for our column chromatography. So next we'll move on to our first fume hood. So here we have our new vacuum pump, which is a high vacuum pump, Edwards. We can get to really high vacuum with this pump and remove residual solvents as well as do vacuum distillations and etc. We have a Schlenk line set up here. So we have a gas and argon nitrogen line where we can dry samples. We can also do inert chemistry. So we can do air sensitive chemistry here in this fume hood. Next we have our second fume hood, where we do our reactions as well. So we have two hot plates set up. We can do multiple reactions in multiple fume hoods at the same time. We also have a mechanical stirrer up here, so we can do large scale synthesis where we just use a mechanical stirrer instead of a magnetic stir bar. And next we have our final fume hood. This is our third fume hood, and here we have our rotovap set up. So we have a rotor evaporator, and we have a couple vacuum pumps in there that we use depending on the volatility. Cooler, so we can use this to cool our any reactions or any of the rotovaps that we need a chiller for. So we have a chiller, and then we have our -20 fridge in the corner where we can store our samples. Next, we're going to move on to this part is still kind of organic because we have our flammable cabinets here. We have two large flammable cabinets that house all of our flammable solvents. And this side is really more focused on analytical chemistry. We have a lot of bench space here where we can do sample preparation, we can do analysis as well. And then we have a second fridge for a fridge, regular temperature and small freezer, again for our samples. We have an analytical balance here and just a regular balance right next to it that goes up to a higher weight. And then here we have our HPLC, and so here we do a lot of work for method development. We did some work on RNA oligonucleotide separation for a company out in Alberta. So there we optimized the separation so that they could monitor two different steps of the reaction. So this is all our HPLC system. We of course have a pH meter because we have to make our buffers and different things for chromatography. We have a second Schlenk line, and then this is all of our glassware that we use for organic chemistry. So that's also having more lab space here, where we can essentially have up to like 12 chemists working at once. So we also have a vacuum oven here in the corner, which we don't have the vacuum hooked up right now. We're just using it as a plain oven right now, but it does have a vacuum vacuum hooked up here. So we can have a vacuum oven. That's where we dry our products, our final solid products. And okay, so this is ARSI's lab. We have 2,000 square feet facility. We have a very intricate HVAC system with 3 different ports. One for the fume hoods, one for the main lab, and one that actually sucks in the gases from the floor. And then we also have the 3 fume hoods, and we also have our data system up here. So this is our server, which stores all of the internal data. So we have a very secure data storage system, and then we have several security cameras located throughout the lab that also ensure the integrity of our work.
So Charlie, what are the services normally you guys do provide?
So we do custom synthesis, we do scale up and process development. So we can take your process, like we replaced some explosive reagents in one of the processes earlier this year. We also do custom synthesis of new molecules that haven't been made before. So we have a project where we made something brand new. We do drug development services. We do analytical services, including method development, formulation. We also do lab services, so if there's any testing or calibration or any validation we can do as well. So we have a wide variety of services that ARSI provides.
Who are the main customers?
So the main customers for ARSI are we have a lot of customers that are other small pharma or biopharma companies. Also university labs often require custom synthesis for their different studies. And potentially we could also service some research hospitals. And but primarily, I would say small biopharmas. And
do you have any collaboration with the universities, and how does that work?
So we do have a few collaborators at the university. So we have two things that link us to the university. So one is that we use a lot of their services. So we use their nuclear magnetic resonance. We use their LCMS, so liquid chromatography, mass spectrometry. We get our mass spec done there. Other characterizations, if we need X-ray crystallography or any complex sort of characterization, because we also do structure elucidation here at ARSI. So we've had a project where we worked out an unknown structure of an unknown for a pharma company, actually a custom research organization. And so we do a variety of different services for a variety of different